Hi everyone, thanks for the organizers to invite me and accept my work for this conference. Are you listening to me? Okay, I think yes. This work is about the analysis of gene expression of one family gene, that is string genes, and the relevance in acute myeloid leukemia. And in this work, we will present a simple strategy to identify a prognostic factor in acute myeloid leukemia also. So, acute myeloid leukemia is an aggressive disease that consists in the block of the hematopoiesis in the myeloid lineage, with a consequence of the reduction of the granulocytes and monocytes and other cells that are measured in the peripheral blood, and accumulation of the blood cells in peripheral blood, bone marrow, or spleen. Because the higher rate of mortality of this disease, it requires precise risk stratification to achieve optimal treatment outcome for patients. Uh, in fact, there exists some molecular and cytogenetic markers to permit stratify the patients in three groups: favourable, intermediate, and unfavorable. The favourable are patients that are treated under standard chemotherapy, and the intermediate is some problematic group because some patients, in fact, belong to the first and some belong to the unfavorable. So the identification of this group is very problematic. And another unfavored group, these patients are treated with intensive chemotherapy or hematopoietic cell transplantation. Also, the prognosis of this is largely determined by these cytogenetic and molecular lesions. More prognostic factors are necessary since less than half of patients respond to well to chemotherapy or stem cell transplantation. So more prognostic factors are needed or are welcome to improve this stratification. In this context, we are studying a group of proteins that belong to the TRIM family. These proteins are characterized structurally by 8 domains. The first 4 domains are highly conserved, and the other 4 are very variable. So this variation permits classify the TRIM proteins in 11 groups. And in humans, we have more than or about 80 members of proteins in function in our cells. The relevance of these proteins in cancer are because are correlated with the stemness of the cancer cells in different mechanisms. For example, we have here in this image several components of the TRIM family in green and in red. They are modulating some relevant pathways in the stemness, like JAK-STAT, for example. JAK-STAT pathway are relevant in acute myeloid leukemia, but in general, in tumors, NF-kappa beta, HSCRUH, WNT pathway are necessary to the stemness in cancer cells. In green, we have three proteins that facilitate or help to the proteins to activate these signals, but we have also some proteins, some TRIM proteins that act as a suppressor tumor and that block this pathway. Not only modulating signaling pathway, these TRIM proteins are relevant, but also modulated some transcription factor relevant for the cancer, for example, SOX2 or CD4 or MYC, that are necessary to the proliferation and stemness. Also modulate genes for the differentiation process and cell renewal, modification of the chromatin structure, and metabolic requirements. So the variety of functionally these family proteins are very interesting in cancer. But in blood cancers, these family are less studied. Some studies indicate that only 3 proteins, TRIN19, TRIN24, and TRIN62, are relevant in acute myeloid leukemia. Specifically, the first, TRIN19, are relevant in a subtype of acute myeloid leukemia, that is acute promyelocytic leukemia. This protein is also known as a PML protein, and in these patients, or in patients with acute promyelocytic leukemia, are translocated with RAR alpha, another gene. But in the other types of AML, only 2 proteins are studied until now, that is TRIN24 and TRIN62, and these proteins are correlated with the prognosis of AML. But how about the rest of the complexity? So in this work, uh, our objective was to explore the impact of the expression of TRIM genes on prognosis of acute myeloid leukemia. Our methodology for that, we use two groups of cohorts. The first cohort, this is a screening data set, that is TCGA. We choose this as a screening data set because it have a complete data of expression by RNA-seq and also have a complete clinical data. And all of the results here in this cohort, we validate in another 5 cohorts. The one first is the VDML. This is a new cohort, have only 2 years maybe. That is also RNA-seq, but another 4 are microarray data. The inclusion criteria for our study are all of the data have to be publicly available data. We did not include a patient with APL because this is an entity different of AML, so it's not included. We consider only patients under standard chemotherapy and patients between 18 and 65 years old. Our strategy to identify TRIM genes with impact in acute myeloid leukemia, for that we dichotomize the patient according to a TRIM gene expression using a cutoff value calculated using the survival rate curve. Okay. After that, to determine the prognostic value, we use 3 criteria. The first, pass the univariate cost regression using the survival data, that is event of death or life. Also, pass the multivariate cost regression. In our model, we include in the multivariate model include a gene expression of one of the 3 genes, age, gender, leukocyte count, and cytogenetic risk. This because there are known or well known prognostic factors in acute myeloid leukemia. Also, we consider that a TRIM gene have ability to predict survival if the area under the curve is more than 0.5. All of genes that pass these 3 conditions in our screening cohort, we validate in another 5 data sets. Finally, we perform enrichment pathways using the gene set enrichment analysis, and all statistical analysis was done in RStudio. As a result, we found that from 63 genes in our screening cohort, TCGA, only 7 demonstrate to have prognostic relevance. Here we have the univariate analysis, and here the multivariate analysis. In the univariate analysis, 4 of these 7 genes act as an oncogene because the higher expression is related with poor prognosis, and other 3 act as a tumor suppressor genes. And checking the ability to predict survival using the area under the curve, we have the only TRIM8 have a best area under the curve, that is 0.65 with a sensitivity of 72% and specificity of 55%. So this is the best prognostic value between these 7 genes. So to validate these findings, we check the prognosis of these that have these genes on AML patients with another cohorts, and we found that only in TRIM8 that we found in TCGA were reproducible in another 5 cohorts. Only TRIM8. So we from 60, we only have that TRIM8 is relevant in AML. Analyzing the survival plot using a Kaplan-Meier method, we have the higher expression of this TRIM8. Patients with higher expression have a worse prognosis in AML using 4 different cohorts from different parts of the world, and also analyzing the area under the curve according to the time. The prognosis value of the TRIM8 gene was strong only in the first 2 years of the disease and the treatment, and is reduced to the 5 years. As was seen in our multivariate analysis, TRIM8 is not correlated with any variable that is known to have prognostic in AML, that is gender, age, and leukocyte count. But we found that some correlation between the expression of TRIN8 with the percentage of blasts in peripheral blood. However, these differences are not clinically relevant. Also, we checked the impact of TRIN8 expression in the event-free survival. This analysis is relevant because in this analysis we include not only the death for the disease, but also the relapse cases. Evaluating the mutations, no mutations in acute myeloid leukemia like FLT3 and NPM1, we found that TRIN8 expression are correlated with the mutations in EDH1/2 and TP53. However, checking the other cohorts, we did not reproduce or we did not validate the results. So the conclusion here is that the expression of these TRIN8 gene are not correlated with any mutations in MEL. And checking for the expression of this gene, in general, we have that TRIN8 are not downregulated or upregulated. But looking for subtypes of AML, we have that patients with translocation 8 to 21 have lower levels of transcript of this gene. Check for the pathways. We have that were enriched pathways relevant in acute myeloid leukemia, that is here, and some pathways that are relevant for the stemness, like JAK-STAT, that is relevant in AML, and also production of interleukin-6. Interestingly, also were very enriched in 3 different databases, that is Gene Ontology, KEGG, and Reactome, that NF-kappa beta and toll-like receptor signaling are very enriched in patients with MEL with higher expression of TRIN8. That's very interesting because there exists some publication that correlate the TRIN8 protein with activation of NF-kappa beta signaling. So our hypothesis is that in MEL cells, TRIN8 are positive regulator of axis toll-like receptor in NF-kappa beta pathway that collaborate with the leukemia by inducing pro-inflammatory microenvironment that is a known condition of cancer progression. So, as conclusions, TRIN8 gene acts as an oncogene in MEL, conferring worse prognosis. The same gene expression are not associated with no MEL mutation, highlighting the possibility of additive effect that with this mutation that we are calculating now. And genes of the axis of toll-like receptor and NF-kappa beta pathway are enriched in patients with higher expression of TRIN8. This is very interesting. And to confirm that findings, we have to perform functional studies in vitro and in vivo to clarify the role of TRIN8 in the aggressiveness of leukemia, acute myeloid leukemia. That's all. Thank you. Sorry, Art. I'm not listening to you. I'm not listening to you. I don't hear you. Sorry. It's not possible for me to listen to you. I have a question here. Can I answer that? Or it's not possible? Okay. Question is, are you able to do pathway analysis to see other pathways are affected by differential expression of TRIN8? Yes, in fact, we had more than 100 pathways, and we select only that what mentioned here because in our opinion are very interesting, but we have other complexity of pathways that were enriched that we have to analyze.
Syndromic X-linked intellectual disability?